Good morning, good evening. Thank you for listening. This is an agricultural or horticultural gardening show focusing on plant science, soil science, and microbiology. The information is fact-checked. You can rely on it, and any of their papers or websites that have been discussed will be put in the description below. Today is a continuation of the Microbe in Agricultural course. So if you're watching this back, fast forward until you see the screen change. That's when the slides have started. Because it's usually at least 90 seconds from now, so give it a bit of time for folk to arrive. Yep, I'm testing. Good. Everything's working good. So it starts in just over 60 seconds. Enough time to run and quickly get a drink of water. Wait for folk like 420 Fred. Hello, 420 Fred. So 50 more seconds and it starts. $35. $25 to go. 20. Quick 5 seconds, wasn't it? All right. So today is a continuation of last week's. So we're going to be going through the slides again. And this week's topic is IPM. So what's IPM? Integrated Pest Management. It combines different ways to manage pests. And because I'm into organics, this will be focusing on organics because there's huge ways you can do it without it, but the organics is very targeted so you can be sustainable. So to continue it here, pests are unwanted insects or animals that damage crops. Instead of relying on just one method, IPM uses a mixture of techniques to keep plants healthy. And so the benefits of environmental protection, profitability, social and risk, challenges in adopting it, lack of awareness, using preferences, technology, inaccessibility, policy and culture. Well, the challenges in adopting are more so getting access to the right species and bios that you need, and having the education to implement them and know when to use them and what to use. So that's what I reckon is really the challenging tech bits. Principles of integrated pest management. 3 ways: natural control, sampling economic levels, and insect biology and ecology. Actually, I'm doing an entomology course on insects this semester, just for this reason. General steps of IPM program development. Prevention, suspension, monitoring, decision making, intervention, and evaluation. So it's a good overall thing of knowing what your crop's doing, what your environment's doing, and a bit of climate prediction, where you can get things like growth, vegetation, density insects, and different other tools that you can use online to help you to predict the season coming ahead. It might be one that's going to be very hot and dry, so you've got to do methods to maybe boost the water or things like that, or it might be an outbreak of something. So it's good. It's good it is anyway. So in this section, there's 4 sections. We'll see what we get up to today. I'm not going to read them all. Some cool ones though. Biocontrol methods will be very good. So introduction. Or like little graphics. So this one's an overview of the plant microbiome composition, complexity, and diversity. So you can see the the endosphere, so inside of the parts above the phylosphere, this is the phylosphere, so inside of the phylosphere, has a lot of colony forming units per gram. Rhizosphere is made up of all these nematodes, the soil microbiome, virus particles, and arthropods. Actually, the rhizosphere is just within, I suppose it is, because those inside of it. The rhizoplane is, maybe not going into that. Synthetic pesticides and fertilizers are not sustainable. You heard it. They'll market and tell you the different things, but they don't know. They're just marketing. They want you to buy their stuff, but it's not sustainable. That's why you go to all these measures of getting the right biocontrol methods and implement them, and things will start working for you. You're not going to keep killing them off. The reason why they've put that in is big marketing people, they've convinced people to grow otherwise, to use NPK fertilizer, non-organics, and then use their pesticides. Then they've got a good business. That's how the population's been educated. And sad. That's why the show's got virtually no viewers, because people aren't into organics, because they're not, the education doesn't go down that route. Marketing's too powerful. So there's health risks, disease development, environmental impacts, and harmful non-toxic organisms. That's a good read if you want to go for that one. You put that into your search engine and it'll come up with those information, because you can see the paper was written in December 2022. So it's just really, it's all to do with microbes, keeping all of the little microbes going that function in the nutrient cycle. Oh, and here's the plant microbiome. How cool, because there's all different microbes everywhere at every different stage to do everything. So there's air in the soil, they transmit it. Private buildings, intensive care units have it. Clean rooms have it. Everywhere. And you notice that there's a lot of different families of microbes here. And why aren't we sick? Because all of the anaerobic ones, the one without oxygen, they're the ones that cause problems. And the ones with oxygen, which is all these ones you see here in the air, they generally don't cause problems. Otherwise, we'd be always sick in that. So by getting good biodiversity and range of them, you're pretty right. So it's these clean rooms that are the problems, because they haven't, if you always in those areas, you're not going to have a good microbiome. You're always going to get sick, etc. This is on microbes in agriculture focus. Yeah, that's a pretty good overall graphic. It just shows you how everything cycles. Everything's involved with everything. Underlying mechanism of soil suppression. Microbes go right back down, right to the culturable level. So what's this saying? Oh, this is culture of isolated microbes. So the illustration shows the microbiota establishes disease suppression of soil microbiome by employing an array of direct and indirect mechanisms. Collectively, these diverse microbial communities perform significant beneficial activities that promote plant growth and sustainable and suppress harmful pathogen functions. So it all works well. Introduction to plant immunity, induced systemic resistance and systemic acquired resistance, competition for ecological niches, microbe signaling, okay, what happens? Selectivities to suppress to give you that result. And then when they've done the lab, they've cultured them up and isolated them, and that's how they get them. And then they do tests on them to see what those different species and individual strains do. Synthetic community development, inoculation to conductive rhizosphere microbiome. Is that graphic again? Biocontrol is a pest management strategy that employs natural enemies or organisms that manage and reduce harmful pests, diseases, and weeds in agriculture and natural ecosystems. Biocontrol minimizes reliance on chemical pesticides, fostering a more eco-friendly and sustainable approach to pest management. So here's where they've put a parasitic wasp to control their bugs in cotton. And I've done it lots, and it works. It targets pests like thrips, Cucumerus targets thrips extremely well, or Stenonemia felti, nematodes, beneficial nematodes, target fungus gnats extremely well. That's like their main diet. So if you can get those types of biocontrol happening, you've won the battle, and you'll find it'll continue to do it for you as well. You just got to see, can it season? Does it cycle, or do you have to keep it warm, for instance, in your compost through winter? Then you can keep it happening throughout the whole growth period over decades. It's great. And to end that too, that's the reason why big marketing people don't want you to do these techniques, because they're going to struggle to make money off you. Importance of biocontrol, sustainable pest management. I'll see if it's even arrived. Oh, Chevro. Hello, Chevro. You know, mate, really nice to see you. Yes. Doing well, he says. All right, struggle on. Back to this. The importance of biocontrol, sustainable pest management. Well, there's less chemical use, way long-term solutions, promotes biodiversity. And when you get all those different microbes happening for you, you might get the ones that will not only do one thing, but those, what's an example? Trichoderma. You might have a problem with, what's trichoderma? What's another example then? Anyway, your microbes might produce phytohormones, which give you an extra bonus. So it's like, you know, when athletes have hormones, they get a pretty good boost. Same thing for the plants. If you can give the right hormone for the right reason, it's going to get that boost. And a lot of microbes, will pretty much all the ones I do use, they all produce phytohormones. And I'm getting some way awesome expressions. Like these, what expressions? Well, my carrots are growing stupidly large. My brassicas have turned into perennials where they just keep cycling, flowing out fruit each year. They shouldn't do that. I've managed to cross broccoli and Brussels sprout and make seeds that produce both of those out of one seed, which even the search engine says it can't be done. But, well, I've got tested 10 seeds and 3 of them come up. Anyway, that's an example. Stuff like that. Biocontrol agents are essential for sustainable and effective pest and disease control management, offering significant benefits in both agriculture and environment. So many rad benefits or merits of biocontrol, like pest and disease, targeted action, enhanced agricultural sustainability, safety and health benefits, economic benefits, reduced environmental impacts, and resilience to environmental changes. Yes, and with this climate change stuff we're in, that's what you want. They help overcome through stress periods. They're really good at stress alleviation. Overview and direct and indirect mechanisms of biocontrol involving between all agents. Okay, just looked at the graphic. Because in a healthy soil, it's 50% plant pathogens and viruses are in there, and it's to do with the beneficial microbes and the ones that are functioning well to keep everything cycling well. So it's when you go and put pesticides on it and spray and kill all the good ones, or at the shop, they don't tell you anything about that sort of stuff, because I'll just say, go and fertilise again, use NPK. Yeah, and you'll go green and grow again. But what about all the microbes that control everything, cycle everything? That's what this focuses on. Because see, they don't just, it's not nutrients. See down here too? Antimicrobial metabolites. They put out enzymes, there's nutrients, they chelate things, nutrients, quorum sensing. So there might be, there's antibiotics, the quorum sensing with the bio films that they put out. Enzymes, yeah. So although biocontrol offers many advantages, it also suffers from potential challenges and limitations, like variability of effectiveness. Well, that's education. Complexity, implementing them. Well, that's education. You've got to put them on at the right time, know their growth and development stage. You don't put mesophilic microbes in at psychrophilic temperatures, because they're not going to grow. Like you don't put normal microbes in at cold temperatures, because it won't function. Potential non-target effects. Well, know your microbes. Work with ones that target good things that are very beneficial. And you're pretty much right, that's hard for that one. Cost, yes. And availability, that's probably the biggest one, isn't it, in education. Resistance development. If anything, they might help with that. Regulatory and legal issues, okay. Yeah, that's for commercial growers. Integration challenges. But see, if you stick to organics, you're pretty much right. So you don't have to worry about legal issues and regulatory stat decks and all those acts and codes. Primary methods of biocontrol, conservation of existing natural enemies. Yes, well, if you can, but most places have been wiped out by pesticides, insecticides that have just killed everything. So it might build an unfavorable pathogen amount, which you don't want. Like I got a big massive compost heap that's massive, and at the bottom of it, it's sitting in a bit of water, and it turns out it's culturing Pythium. Introducing new natural enemies, that's by augmentation. So this is by getting those specific species and strains to do individual tasks for you. That works brilliant, really well. And then it keeps working on and on. It's just, that's the best bit about it. Mass rearing and periodic release. Well, you've got to release at the right time. IPM, it's nearly write it down. You have a big calendar out. And when's the cold coming? When's the spring coming? When's the ground temperatures being affected? And those things are, the temperature is really the most favorable thing. The water content and temperature is all you're really going to know for your IPM. But not all you're going to know. They're the major things, because that's what your microbes cycle on. Conservation of natural enemies. Well, this is your biocontrol. Face wings pretty much do everything. Let me pull up a chart for you. Hey, Genetic Memory Farms, go, mate. Nice to see you. Good evening, peeps. He says, didn't get my notification for some reason. Oh, blimmins. What am I getting for you? That's right. It's under microbes, the IPM chart. Oh, no way. Microbe size. Hang on. Biopesticides. No. Biocontrol. There it is. I got it. You're right, mate. Lucky these are recorded. It's only if you're probably want to ask questions and stuff, and I like things live too. But here's biocontrol. So if you pretty much notice here, for all of these insects, all these predators that are bad, lacewings do the right half, and Montgeronesis do the left half. So by having those two in your garden, holy mackerel, look at that. It takes care of pretty much everything. So that's one example of getting the right biocontrol agent to do the right job for you. For those people in Australia, that's where you get them from. In Canada, I used to get mine from, actually it's a pretty, I think it might be the same name over there too. She used to have everything. There's this lady that used to do it. I didn't receive Montgeronesis though, which is really rad. Those, well, everything. A lot of pests. That's one example of targeting the right pest for the right biocontrol. So conserving natural enemies is a key strategy in protecting our ecosystem. By protecting and encouraging these beneficial organisms, we improve pest control, support biodiversity, strengthen ecosystems, and promote sustainable farming. Natural enemies also provide important services like pollination and natural nutrient cycling, making our agricultural systems more environmentally friendly and resilient to climate change. This inevitably cuts production costs, reduces pesticide pollution, and helps prevent pest resistance. Isn't that rad? So all this information is very, if you're into gardening or horticulture or agriculture, this is just, and you can see it's the latest of information. This slide here is from, the study is from 2024, IPM down the bottom. IPM strategies for citrus, different types of natural enemies. They've got more that weren't on my list. So you might want to source that if you couldn't find stuff, if you being real specific. I always put stuff into my search engine and go from there. And you might have to read a study to get that specific, what's this one? Cocking lid, that one out, because that one might specifically, looks like an aphid looking thing. Sorry, ladybug looking thing. So it might do a certain species of aphid. So that's how you find your exactly what you want. And then once you've got it, you put that name in your search engine and just go buy that and see what comes up. And hopefully you'll find some that you can source that can deliver to you, which is how I do my biocontrol. Providing habitat for natural enemies is a must. All your sticks, your twigs, all those different types of organic matter, there's all the microbe homes. So you can also put, what else to say? Creating diverse environments like hedge rows, cover crops attract beneficials. Here's an example down the bottom. Native ladybugs prey on Colorado potato beetle eggs, but also rely on aphids and pollen. Through further research is needed to optimize their use in pest control. Introducing new natural enemies, classical biological control by releasing non-native species in a naturally controlled invasive species area. So it's, a lot of these things have been shied upon because people with bad education, or you go to a shop and they're been sold by their sales rep. So yeah, if you sell 20 of these, we'll give you a kickback of a holiday. So they just sell this stuff to you, and all of a sudden you're getting it, and then it destroys the environment. So you have to have a little bit of background education into it. Don't go trusting no sales rep in these shops. So yeah, identify your pest. So you take a photo and go to your search engine, put the image, image search, put it in, and it'll come up with pretty much what you got. Then you know what you're working with. Then you can put in, I told you how to find it, and quarantine and testing, access the potential natural enemies to ensure they won't harm non-targeted species. Oh, this is full on. Rearing, release, and monitor. Okay, that's if you want to change an environment. Or maybe introduce something from overseas into your natural environment, which is how the government would test things. Because quarantine and testing, that's right up there. It's going into that. Precautions. Successful examples including controlling the alfalfa weevil with introduced parasitoids. So they've introduced a biocontrol agent that's worked really well. They have to see what the adverse effects of that in the environment are too. Might have bad problems like the cane toad in Queensland. They introduced that, and that's just gone, and for the sugar cane, and that's just gone and destroyed a few other things. There's the reference down the bottom. IPM World Textbook 2024, Radcliffe's mass rearing and periodic release. This is what the plate companies that you buy off do. That's how they can sell you the numbers that you need, the millions and billions of numbers that you need. So I'd be real careful about doing this, because you might really create a big problem. You don't want to overpopulate an area that just might create problems. So I hope you're picking up what I'm putting down. You want to just get a little bit. Like, they'll sell you a big container and it'll say this will do an acre. Okay, good. That'll do my acre. You might need 3 acres, or 20 acres. So that's what I'd more so go with. I wouldn't go with this method for that reason. It might wreck the environment. Let's just say BT. Biological insecticides are rad, like BT. I use BT and beneficial nematodes to target support pests without creating lasting problems. Yeah, I'd like to, that's an example actually, just saying don't do it. What I'd like to do is BT is one of the actual ones that's really rad, and it produces indole acetic acid too, a auxin phytohormone. In something release involves mass rearing of natural enemies such as ladybugs, and then put them in with them. Righto. Okay. So that's a job. That's actually nearly a job if you want to get into it, because it's relatively not new, but it's sort of new. Not many companies are doing it. So if you want to make money in the agricultural sector, this is one way where you lemon can. And everyone's going to need it soon, because the world has to change and be more sustainable. So they're getting off this herbicide, pesticide, right, and they're going to the biocontrol agents. And biocontrol agents are living organisms, such as microbes. They target pests and pathogens in agriculture. They do the work reducing the pest and pathogen population. It's rad. They are a key part of IPM, providing eco-friendly alternatives to chemical pesticides. Examples of trichoderma. Doesn't say what they target. That's what they are. Yeah, we all know what they are. There was a chart in last week's one which had an example of target. Actually, I think this one's got it too. Yeah, I'll show you. Biocontrol agent. Oh, gee. White fly. No, I want to buy actual biopesticides. Kind of wanted to biocontrol more the bugs and insects, but this does come under it because it's controlling with bios. There's an example. I'm not going to read all them. I went through them a few shows ago. Current to 2023. Yeah, isn't that? You can also get ones for weeds too. Pretty rad. Spinosad. Back to this. Types of biocontrol agents. Oh, really? I just had to go to the next page. So they come in 4 categories: predators, parasitoids, pathogens, and competitors. A predator example is like a ladybug, which actively consumes aphids. Parasitoids, which kill hosts and by laying eggs inside of them, entomopathogenic insects. And a pathogen is a Bacillus thuringiensis, which kills and infects them. And competitors, like Pseudomonas fluorescens, compete with pathogens and cover crops, compete with their weeds. Also, Pseudomonas fluorescens is rad for Oomycetes, which is a Pythium. These agents are key components of IPM. Well, as an example, if you know you're going to get a big outbreak of something I was saying before, you might want to go and get some parasitic wasps, or refer to that chart that I put up before. Types of biocontrol agents. Just goes into a little bit more detail. I'm not going to read them now. Press pause if you'd like to read them. I covered them before. I'm not going to go into great detail. Predators. Specialized predators or general predators. So specialized ones are ones that target specific types of pests, kind of what I was going on before. But for the general ones, like Montgerensis or lacewings, they are good for just overall. They eat heaps, and they're good overall. There you go, 2 examples. Some predators. More examples, like the rove beetle, hoverfly, silvergrid fly, or the pirate bug. Like the rove beetle is good for small insects, like root maggots, onion maggots, cabbage maggots. Hoverfly doesn't say what it's good for. And pirate bug is good for thrips, spider mites, aphids. These bugs can eat actually 30 spider mites a day. Pirate bugs. Parasitoids are insects that lay eggs, their eggs inside another insect. For good reasons. Endo inside, ecto on the outside, wherever they lay it. The 2 main types of parasitoids. Is an example. What's this bioagent? Aphidus colomani is a spore parasitic wasp that attacks many types of aphids, and it's mostly active in the warm temperature. There's an example. So if you're thinking, oh, I've got heaps of aphid problems and it's, gee whiz, what do I do? But it's still in cold temperatures, getting frost. Well, this isn't your answer. This isn't going to work for you. Why the temperatures are real important to know how it functions. Like it might work in cold temperatures, it might eat 2, or it might die. So that's, in my research that I'm finding, I'm putting for different microbes, the specific ones to temperatures, their growth and development. How do they cycle? How do they do their functions? And endos ones are Encaria formosa of nymph of greenhouse whitefly. So it's just doing the same thing, laying the eggs on the outside into the nymph on the underside of leaves. Pathogens. Pathogens are tiny organisms like bacteria, fungi, and viruses that can make insects sick. Yeah, my batch culture, spirulina, that had a fungal infection a while ago. My bacteria had a fungal infection a while ago, and I got rid of it, but it was just sort of funny. I'm glad that I got rid of it. Disease. Here's an example: Trichoderma species which effectively control root rot in sunflower caused by fusarium. So if you go and get Trichoderma, if you got fusarium, and if you get Trichoderma hazarium, it's not going to have, it's going to kill fusarium, but it won't have oxen produced by it as well. Where if you were going to source the species of Trichoderma variety, that produces oxen as well as kills fusarium. That's an example of targeting the right species and knowing a bit about it to get the bonuses of the phytohormones as well, which is the route that I've gone down and giving all these weird things. Pathogens are many insects in one area. Disease can spread quickly among them, especially in the environments right for the pathogens to thrive. Microbes work in mechanisms such as competitor antibiosis suppression and hyperparasitism. Okay. Fungal biocontrol is used to control harmful pests and pathogens. Fungal can either directly attack pests or pathogens, offering a friendly alternative to chemicals. Like Trichoderma, went through that before. Bavaria bassiana, entomopathogenic fungi that kills a lot of insects. It's rad. Metahizium, similar. That just gives the Mescadie disease that it produces through these two. This one's green, this one's white. Another example down the bottom. Clyogladium virens, which I haven't worked with. I've worked with all them. I work with. They're in my IPM. Oh, some more. Candida. I know about that one, but haven't worked with that yeast. What's it? Oreobasidium polarins. This diatomaceous fungi combats pathogens like botrytis and penicillium. Fair income. The grey rot that goes into the carpels and the flowers, the fruiting areas of the plants that you'll see, the grey rots, botrytis cinerea. I've never worked with that one. Geez, I love doing re-looking over this stuff. See, that's something that I could look up and try and source that. I would like to. Candida. The yeast prevents rot in citrus and apples. And helps against penicillin species that are harmful. Other yeasts, such as Saccharomyces cerevisiae and Pinchera anonoma, also produce antimicrobial compounds and enhance plant defenses, making them promising alternatives to chemical pesticides. Yeah. They produce antimicrobials compounds, but you've got to also think then too, does that affect my good ones? Because I've looked at Saccharomyces cerevisiae available everywhere, and I thought, how can I use this to my benefit? I haven't really found a benefit. So it's like getting that and implementing in my IPM, that might kill off and be competitive with other microbes. So make sure the ones that you use are ones that you want. Advantages of fungal biocontrol agents. Eco-friendly, target-specific, reduce resistance, soil and plant health, IPM. My goodness, gone a bit backwards there. Virus and bacterial phages in control. It's another way if you've got really a hard problem, trying to, if it's you've tried all these other methods, this might be the last method. Like tobacco mosaic virus, TMV, for instance, there's a bacteriophage that you can get that works specifically. It has a strain against that. So that's where you'd source that. If you had a big commercial crop and I was going over them, they've said, we've tried this and this and this and this, I'd be going down this route. implementing a bacteriophage or a beneficial virus. Oh, now they give examples too. I don't go through this before anyway, here's a PRSV, the papaya ring rot virus. There's a mechanism that you can get. They've gone and done the test, just like I was saying. A specific strain that's derived in Hawaii to fight this. Pretty cool. Agricultural is rad. So you don't need all this pesticides, herbicides. You just need a little bit of education. Here's another example. Hypovirulence virus that affects the fungal pathogen sclerotinia, which is a white mold disease in various crops. Pretty rad. Here's bacteriophages over the counter. So bacteriophages are viruses that infect or kill specific bacterial pathogens. What have you gone about? Here's one there. Agrophage. So you probably find by contacting the person that sells that, they don't really know much. They don't know that it gets like Xanthommonas species, for instance, that targets. So it's hard to trust what people are saying. So that's where I write to the companies and ask them. Actually, I've been talking for a bit. I don't want to go through too much. A few words here. GMF says, just to catch up, is all of this information on application, just as application applicable indoors in greenhouses and outdoors? How do you keep them where you want them? They cycle. So they'll only attack pests. So if you go and get ladybugs, because you've got an aphid problem, once all the ladybugs go, once all the aphids go, a lot of the ladybugs will fly off and they won't continue to breed. But you'll get some that will stay there. So by keeping that on, that's going to give you some ongoing protection. So that's out in the field. In a greenhouse, it's just easier to control. But I'm talking general field specifics, because indoors is just easy to control. You just get them and they do the numbers and that's it. They die off and you're happy. But outdoors, you want it to be sustainable and continuing. So, and then you'll find that some of the biocontrol and microbes, they'll actually go into your compost and they'll be in your soils and they'll be cycling through that, like your trichoderma species, trichoderma, not grammar. Your Trichoderma hazarium or Trichoderma variety, for instance, they'll be in your species where you cycle in your soil substrates. So you might scoop some out and put that in your compost, or you might be finding that these species will be on the outside of the crops, and then that plant material will go into a compost heap, and those will keep culturing up. So that's how you'll keep them going forever, from your fungi and your bacteria and your biocontrol agents. Hope that helped, mate. And temperature. If ones that die off in temperature, for instance, Monterenesis isn't good for frost. So if you get a heavy frost, bang, they're dead. That's where you'll have to know that and get them next season. So when everything thaws out, spring thaw comes and you'll be buying them again. So that's why you'll be writing down all these IPM methods. Temperature, very important. I would love to be a bug rancher. Says GMF. Mate, I reckon it's rad too. It interests me. There's big monies to be made in it. I'll help you. I'll individually help you. And my course this semester is entomology, which is pretty much on this, on how to culture your own specific ones. So I'd help you, mate. There's mace. Hey, Mace, how you going, mate? Nice to see you. GMF says, yeah, Saccharomyces makes alcohol, so it kills and competes with everything except Acetobacter. Yeah, see, you got to be careful, haven't you? Got to be careful with what bugs you put in for the right times to do the right job. I'll just finish up here with this. It seems like there's a few more to go. Quick questions in chat if you have any. Traditional approach in biocontrol. Let's just keep going. Oh, up to mechanisms. Oh, whatever. Mechanisms of action. So understanding the mechanisms of action is crucial for developing effective formulations and obtaining regulatory approval. Despite challenges in studying complex integrations, the four main mechanisms have been put down here: antibiosis, competition for nutrient and space, parasitism, and induced resistance in host tissue. Here's the different modes of action. So all those things we've already discussed, you can implement them, it says. But remember, it's temperature, big thing. Just remember that one. And bacteria that produce antibiotics will inhibit or kill bacteria and fungi and serve as a highly effective biocontrol agent. Here, Subtilis, Amyliciformis, unknown, Ligoniformis, Megatherium, Cereus, Brevius. I use half of them. So what's this saying? Number of strains. Yeah, good. What do? The number of strains in each species and the modes and the mechanism of targets of action. 2022 study. That'd be a rad study to read. So cell wall, cell membrane, intercellular process, and quorum sensing. Quorum sensing is when microbes speak to each other because they've got to talk to each other. So that's what quorum sensing is. They put out in their biocontrol the chemical signals that tells the bacteria what to do, what to produce, etc. That's quorum sensing. So Subtilis, they all know Subtilis is actually beneficial for humans too, as well as plants and soil. Amy is very expensive. So is Ligoniformis and Megatherium. Cumulus, Brevius. Okay, good. What's this? Number of strains. What's it? A and B. A is the number of strains in each species. Oh, okay. Because there's been heaps of studies being done on Subtilis, Bacillus subtilis, so that's why they've got 73 strains. So what do you want to target? Well, I want the human body to be better, be skinnier. Well, we've done a study with Subtilis, and it found that happens. You need strain number this. Oh, cool. Or we want Subtilis to take care of, it's supposed to work with this botrytis scenario. So we want it to, what one specifically does that, and they'll have the number. So the 73 studies been done on that. Antibiosis. Targets different areas. Not going to go into them. Competition for nutrients in space. Microbes compete for nutrients in space, limiting their growth. Yep. Example down the bottom. Subtilis, strain number that. SRB28 colonizes, it does a specific job. Same with that one. Pseudomonas genesi. RU47. So this is my area of study and area of interest at the moment. Specifics that do specific, getting specific bacteria and fungi to do a job for you. And it bloomin' works. Parasitism is a competitive interaction which one organism derives nutrients from another. Then there's hyperparasitism, when the host organism is a parasite, such as a plant. An example of bacterium, no. Types of hyperparasitism in fungi, or biotrophic mycoparasitism. Feeds on living host fungus, drawing nutrients through specialized structures called heustoria without killing the host. And then necrotrophic mycoparasitism feeds on them, kills host cells and feeds on them, as well as other organic matter, making them more suitable for commercial biocontrol. More examples once you get right into it. Prominent mycoparasitides in biocontrol. There's some more examples. Trichoderma and Clonostrachys are the most studied mycoparasites, primarily targeting plant pathogens. They produce infection structures and kill host cell walls, degrading with enzymes and secondary metabolites. Some gene specific, very good. Introduction of resistance. Plant growth promoting microbes act as biocontrol agents by reducing plant pathogens and enhancing growth. They contribute to nutrient cycling and increase plant biomass through various mechanisms. An example is Hazarium, which shows antifungal properties. But if you go and get member variety, it'll do exactly that. Plus, it'll have your phytohormone IAA. PGPR, nutrient uptake by nitrogen fixation, phosphate solubilization, cytophore production, and hydrogen cyanide production. They don't all do that. They can do that. So you've got to get the right one to do that. There's some that do two, like what's my favourite one? Bacillus, come on. It's Paleo Bacilli. What? Anyway, that one does two. It does nitrogen and phosphate, but it's very rare. Come on, what is it? I'm going to have a look. Polymixer. This was polymixer. Introduction of resistance mechanisms. They promote plant health without directly attacking pathogens. So these type of agents can help with induced resistance, systemic acquired resistance, and systemic resistance. To be honest, most microbes actually help with that. Yeah, and that's a good argument for someone to prove me wrong, because the FL22 receptor on a plant, of the 30-odd thousand genes it has, that detects flagella in microbes. So once it's detected, it can activate any of these pathways. Induction of resistance mechanism. Priming. It sensitizes plants for a quicker response to prime the genes. So if you use these microbes, breed, you'll prime the genes so they'll have a quicker, a faster mechanism response to problems next time. I've done that for a while. Here's a selectable bacterial biocontrol agents for plant diseases chart for you from 2022. There you go. Screenshot that and save that one. Hang on. And save that as one example. Oh, hang on, no, I've got to pull a year up. The year is very important. Now screenshot it. Because down the track, when you get into your science more and want to learn more about specific bacteria, you'll come across this chart and go, oh, wow, I can get Pseudomonas fluorescens EPS 622E. Oh, and I wouldn't believe I'm working with pear plants at the moment. This is so cool. So that's just having these tools in your kit is so vital. And this one should be in my kit too, which it isn't. it's got strain numbers. To have a chart with all different ones of strain numbers is pretty cool. See, I use Florence, but I don't know what strain number it is. Streptomyces, rad. And the best thing is the disease reduction application amount in colony forming units. So you got to get 10 to the power of 7, 10 to the power of 8 for it to be effective. Where a lot of people will sell them, I've seen them being sold 10 to the power of 4, 10 to the power of 3. It's not going to do anything. Nematode suppression. Microbes effectively can be managed by nematodes. Yeah, we all know that. Like Stenonym felti. I'm going to stop there. We're going to leave next week to traditional approaches and biocontrol. Pure culture methods. Here you go, GMF. This is going to be how to reproduce them all. Ooh. We'll wait till next week. That's rad. Today was a great show. What do you think about today's show? I reckon it was good. That's about it. Still got 4 minutes. Someone wants to chat. Say something. Press the like button, please, if you like this. Share this with your friends and share it on different streams if you can, please, because I would like to get some more numbers on the stream. That's how you can help me. And for those people that subscribe, pay subscription, thank you very much. That does help, even though it's not much, but it still helps in the long run. I get nothing out of this effort that I put in. Sharing all my latest research in agriculture. So fun. It's all rewarding. Well, I find it rewarding. Get the best fruit and veg, organics. Oh, it's just great. Build a soil. All right. Well, I'll leave it to for another 60 seconds or so. It might have been an ad. That's why the numbers drop down. It's hard. I don't push the ads or anything. YouTube just has an ad whenever it wants to. So I find that all of a sudden I'll be looking at the numbers. Oh, well, there's one person watching, but there might be an ad on. So, and this course will end in a few weeks, so I'll be looking for suggestions to do something then. I don't know what else to do then. My uni starts up in a month, 6 weeks' time. Which I can't wait. It's good fun. More agricultural stuff. Latest agricultural stuff. I study in India, and their soils is already stuffed, or a lot of it is ruined from NPK marketing and fertilizer, not organics. So all these courses that I take, they're all predominantly organic based and telling you how to be sustainable. That's the bit I like about it. So we can make our soils or substrate better than when we first started. How cool is that? So especially if it's on your own property, you'll be building your soil to be better. So when you leave, you'll know you've done the right thing by Mother Nature as well as keeping everything cycling. So it's very rewarding. Organics. I find it rewarding. And it's getting easier and easier. Now we've got these search engines we can just put stuff into, find out what our biocontrol agents are. Gosh, it's just that sourcing them is the problem. There's only a few people actually selling them. Finding out with a lot of organic stuff. I asked in a producer, a shop in Australia, make an agricultural shop, why haven't you got more organic stuff? The response was, organics is new to Australia. Yeah, there you go. Niche markets everywhere. if you want to try and make the most of this future that everything's going to be in 10 years, get onto it right now, because you can get your foot in the door. There's going to be so much opportunity for money to be made, which isn't done at the moment. I even tried to source some NPK fertilizer that's organic. It's not around yet. Well, they have got neem cake, $100 a bag, so expensive that it's just not sustainable. So there's good future in all these different things that I've been going through. Let's go. Righto. Put something in. It's been an hour now. Put something in chat if you want. Please press the like button and share this with your friends. That would be good. It'd be nice to have 20 or 30 people and heaps of discussion. So it'd be nice to have like every second or third show to have just a open show where it just be questions only, and there's 10 or 20 questions. That would be so cool. But it's not a perfect world, and I'm appreciative to get any viewers. As long as this helps, I know it'll help someone. It's really good information. I hope it helps. Write in the comments if it does, and if you've got any questions, also put in the comments. If you watch this after the stream, I'll still help you after the stream too. So if you've got any specific problems with areas you can't combat, I could help you. Or I'll try and help you anyway. All right, that's it. That's the show for today. Thanks for listening. Hope you can join me next week, where we'll continue on with the IPM culturing and strategies to make it. And happy breeding, happy growing, good health to you all. Bye-bye.